Hi everyone, this is Ms. Shim, and today we're going to be talking about the cell cycle and mitosis. For my students, this is going to be chapter 12 in the textbook, but the main topic today in this video is going to be mitosis and the cell cycle. All right, let's get started. On this first slide, you can see several diagrams and pictures showing mitosis for several purposes. At the very top there, you can see a zygote that has just recently become fertilized by a sperm cell, and it's going through mitosis. It's dividing into 2 cells, and then from 2 you get 4 cells, from 4 you get 8 cells, and you develop into a little embryo. Over here, you have an amoeba, which is a single-celled eukaryote, and it's dividing by mitosis for the purpose of asexual reproduction. And then over here you have a tree. It starts off as a little seedling and it's developing by mitosis. It's getting bigger, it's getting thicker, taller by mitotic division. And then here you have a lizard and it's repairing its tail. It's lost its tail probably from a predator, and it's growing its tail back by mitosis. So by the process of mitosis, you have gone from an egg to a baby. Originally, you were a fertilized egg that divided by mitosis. You divided, and you were a little embryo and then a fetus, and then you were one day born. So before a cell divides, it needs to first organize the genetic material first. We need to ensure that the 2 new cells, when a cell divides and forms 2 new cells, it needs to have the exact same genetic material copied. And so we need to talk about the nucleus and the cytoskeleton. The nucleus is where our DNA is kept, right? And you can see in this diagram, this is our nuclear envelope. It's a double bilayer. And inside you have the DNA, and it's loosely coiled into this chromatin structure. But during mitosis, that chromatin is going to condense even further, meaning like it gets tightly coiled into a structure called a chromosome. The cytoskeleton plays a really important role in mitosis because the cytoskeleton produces these microtubules. Remember, the cytoskeleton is made up of a few filaments, but the microtubule filaments are the ones that are responsible for dividing the genetic material equally so that the 2 new cells will receive the same genetic information. And you can see in this picture, these are what we call the spindle fibers. The spindle fibers are made up of microtubules. The microtubules are being synthesized and being made at the centrioles, and they're growing longer and longer. And what they do is they latch on, they attach to the chromosomes, and they move the chromosomes around in this cell, and they ensure that the 2 new cells are getting the same genetic material. So in the nucleus, you have the DNA, right? And the DNA is going to be surrounded by the nuclear envelope. And the nuclear envelope is a double membrane. So we're talking a double phospholipid bilayer. And it also has pores in the membrane. And the pores are going to enable exchange. Like what's made in the nucleolus are ribosomes, and the ribosomes are not found in the nucleus, right? The ribosomes are synthesized in the nucleolus, but they have to come out, and they're going to be floating in the cytoplasm, or they're going to be attached to the rough ER. Later on in the future chapter, we're also going to be talking about transcription. Transcription is where genes are copied into an RNA molecule, and those are going to be made in the nucleus where the DNA is, and then they're going to exit out of the nucleus into the cytosol where the ribosomes are going to synthesize the proteins from that copied gene. So once again, the cytoskeleton is going to play a really important role in ensuring that the 2 new cells receive the correct genetic material. The centrioles, which are an organelle inside of the cell, what they do is they produce these microtubules that are going to form what we call the spindle fibers. And these spindle fibers are going to guide the chromosomes in mitosis, making sure that the 2 new cells receive the correct genetic information. So when a cell divides, what is being passed on to daughter cells? Well, when you have a cell and it divides into 2 new cells, you have to make sure that each cell is getting the exact same genetic material or DNA. You also have to make sure that the two new cells are getting all the organelles that they need to grow and develop, right? You got to make sure that they're getting the mitochondria, right? You got to make sure they're getting lysosomes, Golgi, ER, ribosomes, etc. Cytoplasm has to be split up. Cell membrane, you have to have enough cell membrane to make up the boundary of each cell. Enzymes, proteins, whatever is needed inside of these two new cells, you have to split into the two new cells. That's what's being passed on from mother to daughter cell. So here's just an overview of mitosis. Mitosis is actually, you've got 5 phases, okay? Prophase, prometaphase. Over here, this is called metaphase, okay? Metaphase, M for middle. The chromosomes are, you can see they're lined up in the middle. This is what we call anaphase and then telophase, okay? But before mitosis, we actually have a phase called interphase. Okay, so we got to talk about interphase. So before mitosis, we have interphase. And interphase is basically where the cell is spending most of its life cycle. Like 95% of its life cycle is in interphase. And this is where the cell is doing its everyday job. It's synthesizing proteins, it's doing what it's supposed to be doing. Your cells are what we call differentiated. They're all doing different jobs. And so this is the phase where the interphase is where the cell is doing what it's supposed to be doing. And we can see in interphase that it's basically a timeline of what's going on inside of a cell, okay? An interphase can be divided into 3 different phases. We have G1, S phase, and G2. So you can see here the interphase is broken up into G1, S phase, and G2. In G1, the cell is basically a brand new baby cell, okay? So at this point right here, this is like the birth of a cell. So this diagram right here is what we call the cell cycle. This is the cell cycle. You can think of the cell cycle as being a timeline. This is our birth, the birth of a cell. So G1, I like to think of it as being like the first growth phase. It's just growing, right? It's growing. And then in the second phase of interphase is called the S phase, S for synthesis. And what's being synthesized is DNA. Okay, DNA is being copied because in order for a cell to divide, right, into 2 new cells, you have to make sure that each new cell gets the exact same genetic information. So the S for synthesis. And then next we have G2. G2, now the cell is really big. It's growing even bigger and it's getting ready to divide. Okay, it's producing more organelles, it's producing proteins, it's producing more cell membrane so that it can eventually go through the next cycle, the next phase, which is called mitosis or the mitotic phase. Visually, what's happening here? Okay, so we have a brand new cell. And it has 46 pieces of DNA. Okay, in a human cell, our body cells, we have 46 pieces of DNA. 23 is from your mom and 23 is from your dad. And in the first phase of interphase is called G1. The cell's just growing. It's growing bigger. So I'm going to just draw the cell growing a little bit bigger, 46 pieces of DNA inside of the cell. And then in the S phase is where the DNA is being copied, okay? And all 46 pieces are being copied until at the end of the S phase, you now have 92 pieces of DNA, okay? Right now I'm just calling them pieces, but they're actually called chromatids, okay? By the end of the S phase, you're gonna have 92 chromatids. And then the cell is still growing in the S phase, the G2 phase. It's growing bigger and bigger and it's getting ready to divide. And then in the mitosis, that's where it actually divides, okay? The cell is going to go through cell division. Cytoplasm is going to actually divide in cytokinesis, okay? So at the end of the G2 phase, you have 92 pieces of chromosomes, and then it's going to be ready for the M phase or the mitotic phase, where it goes through mitosis, That's where the cell is dividing. Cytokinesis is where the cytosol is actually dividing. So at the end of the G2 phase, our cell is really big and it's ready for mitosis. It's ready for the mitotic phase. The mitotic phase includes mitosis, which is cell division, and cytokinesis. Cytokinesis is where the cytosol splits, okay? So in mitosis, our cell is stretching and then it's eventually going to divide into 2 new cells. So we have 92 pieces of DNA in our cell at the end of G2, but then it's going to split up into 46 and 46 and then 46 and then 46. And then when cytokinesis happens, the 2 new cells have 46 and 46. So that's how we go from, 46 to having 46 and 46 pieces of DNA during mitosis. Okay, you got to have the S phase. This is so important. The S phase of interphase is where our DNA is being copied so that the 2 new cells have the exact same number of chromosomes, the exact same genetic information. So that's the cell cycle. The cell cycle is a timeline of the cell being born, growing, DNA is being synthesized, and the cell's getting ready to divide in the mitotic phase. Okay, so that is the cell cycle. And most of our cells are actually in what we call the G0 phase. Okay, G0 is what we call resting, where the cell grows and it exits the cell cycle. So here's just another different way of drawing the cell cycle. So, this is the point where the cell is born, right? The G1. But then, so those cells, they never go through the S and the G2 and M phase. They go into what we call the G0 phase of resting. So a lot of our cells are resting. They're never dividing. So for example, our liver cells, our brain cells, muscle cells, they grow and mature, but they never divide. Okay, so brain, nerve cells, and muscle cells. Liver cells are in G0. However, liver cells are very unique in that they can go into the G1, back into the G1, and then the S phase G2, and actually divide. So if you ever donated part of your liver, it could actually go back. It's in G0, but then it would go back into G1, and then into the S phase and G2, and then mitosis and grow back. So interphase is where our cells are spending most of their time in, because you've got the G1, the S phase, and the G2. What's happening in interphase, besides what, it getting ready to divide, you can see that the DNA is in what we call chromatin format. And you can't tell from this drawing or from a picture that the DNA is copied in the S phase because, it's just kind of, it looks like kind of a mess with the DNA. It looks like spaghetti, okay? But if it's gone through the S phase, it's actually doubled the number of DNA fragments, okay? So DNA is loosely packed into what we call chromatin fibers, and the cell's getting ready to divide. It's getting ready for mitosis because you can see that the centrioles have replicated, okay? You can see 2 centrioles. There's a pair of them, and they're going to form the spindle fiber that's necessary for mitosis. So that's what it looks like in interphase. So in the S phase, right, of interphase, the DNA is being copied so that the daughter cells get a complete identical copy. All right, so before we talk about DNA organized into chromosomes, which we'll see in mitosis, in interphase, our DNA is not naked, okay? When I say not naked, that means that our DNA is not alone, okay? Because our DNA is wrapped around histone proteins. Okay, so these are the histone proteins. With prokaryotic DNA, like DNA found in bacteria, they don't have any proteins associated with their DNA. Their DNA is what we say naked, okay? But with eukaryotic DNA, our DNA is wrapped around these histone proteins, and it folds further, it folds even further, and forms these loops, okay? And at that point, we call it chromatin. Okay, loosely coiled loops of DNA and histone proteins. And then in prophase, which we'll talk about soon, in mitosis, the chromatin, it condenses even further, like it folds even further, it coils further until it forms these compact structures called chromosomes, okay? And that's what we're going to be talking about next, what chromosomes are. So after DNA duplication, the chromatin condenses. So you could see here, okay, this is what our cells look like in interphase. But then in prophase, which is the first, phase or step of mitosis, you could actually start to see chromosomes forming, okay? And so that's where the DNA wrapped around proteins is condensed even further, okay? It's you can actually see the chromosome bodies from after the S phase. So that's what we're going to talk about now. So, we have 46 chromosomes, which I mentioned before. 23 is from your mom and 23 is from your dad, okay? And the pairs, the ones that come from mom and dad, they're in pairs, and we call them homologous pairs. Homologous because homo means same, logos means information. So for every chromosome, you have two, and on the chromosomes, you have basically the same information. Like let's say the gene for like eye color is over here. You have one from mom, one from dad. And then in the S phase, okay, in the S phase of interphase, the DNA gets copied. Okay, so here you have the single-stranded DNA, but then after the S phase, the DNA gets copied, and now you have two of every chromosome, and we call these two sister chromatids. Okay, it kind of got cut off down here, but each one, okay, this is a sister chromatid, okay, and this is another sister chromatid, and they're copies of each other. All right, and they're joined here, and they're joined right here in what we call the centromere. Okay, the centromere. Okay, so this is a chromosome. This is also a chromosome. They're just different. They look different because of the S phase, because DNA was copied. So this is what we call a single-stranded chromosome, and then this is what we call a double-stranded chromosome. So this is before the S phase, this is after the S phase, but they are both called chromosomes. One is single-stranded, the other one is double-stranded, okay? So to help us understand what's happening in interphase in the S phase, okay, to visually see what's happening with these homologous pairs and double-stranded chromosomes, I'm gonna draw, let's see, let me draw this first, okay? So I'm going to draw the cells splitting. So we have the mother cell, okay, and it splits into what we call the daughter cells. So here's the chromosome from your mom. So then here's the chromosome from your dad, okay? And then when the DNA gets copied in the S phase, right? Okay, let me get the red. I'm using just the traditional colors for mom and dad chromosomes. Then this sister chromatid is going to go into this cell. Okay, and then this sister chromatid is going to go into this cell. And then this sister chromatid from your dad is going to go here. And then this sister chromatid is going to go into this new cell, okay? And so do you see how the two daughter cells look exactly like the starting cell, the mother cell, okay? So that's why it's really important for the cell to go through the S phase to copy the DNA so that the two new cells have the exact same information, okay? So each single chromosome, it's called a single-stranded chromosome, each one, but then when DNA is copied, we call it double-stranded DNA. And then when the sister chromatids are going to split up in mitosis. Okay? All right, moving on. So mitosis has 4 phases: prophase, metaphase, anaphase, telophase. You do need to know what's happening in each of the phases. In prophase, this is where you start to see, right, the chromosomes forming, because before prophase, before mitosis, in interphase, you have chromatin. You also start to see the centrioles, the double the two centrioles moving to opposite sides of the cell, and then the mitotic spindle forming out of the centrioles. The mitotic spindle, again, are the microtubules that are going to eventually attach to each of the chromosomes and split them, maneuver them around the cell, and make sure that they go into the two new cells. And then you will also see the nucleolus disappear, and then you're going to start to see the nuclear membrane break down, and you can really see that in prometaphase. This is prometaphase. The nuclear envelope is now nearly gone, okay? And the spindle fibers, they start to attach to the chromosomes, all right? And then they attach to the chromosomes at what we call the kinetochores. Kinetochores are proteins that are attached to the sister chromatids. So if I were to draw, like, here's my chromosome, then the kinetochore proteins, there's one attached to each sister, and microtubules will attach to the kinetochores like so, okay? In metaphase, the nuclear envelope is completely gone, okay? And that enables, you gotta get rid of the nucleus so that the microtubules can latch on to each chromosome and move them to the middle, okay? Metaphase, M for middle. Meta is middle, right? And now you have the spindle fibers attached to each sister chromatid, and some of these non-chromosomal microtubules, what they do is they actually stretch, they grow longer, and they stretch the cell in this direction and that direction. They basically are going to cause the cell to elongate. It's going to become a really long egg-shaped cell so that it's eventually going to split right down the middle, okay? And then in anaphase, that's where the sister chromatids actually split up, and the microtubule here just gets shorter, okay? The microtubules get shorter and shorter, and as they get shorter, they end up moving the sister chromatid in this direction, and this one's going to go in that direction, closer to the centromere, closer to the centrioles here, and then you're going to end up the cell's going to end up splitting right down the middle, okay? So anaphase is where the sister chromatids are going to separate. In anaphase, the proteins that hold the sister chromatids together at the centromere, they are inactivated, and then the sister chromatids can actually come apart. So now you have two chromosomes. Each one is a single strand. You started off with a single chromosome that was double-stranded, but now you have two chromosomes that are single-stranded, moving in opposite directions so that the two new cells can have the exact same information because the sister chromatids are identical to each other, right? And then the last phase of mitosis is called telophase. Telophase is where you have the chromosomes now completely on the opposite sides of the cell, and then now the nuclear envelope starts to reform around the new sets of DNA, the new sets of chromosomes. This spindle fiber, it disappears, okay? And then cytokinesis begins, okay? Cytokinesis, we'll talk about that now, is where we have the cytoplasm splitting. The cell membrane is going to split right down the middle, and that's cytokinesis. In animals, a cytokinesis is visible, okay, when a cleavage furrow forms, okay, cleavage furrow. The cleavage furrow forms when actin filaments, okay? So we've talked about microtubules, which is part of the cytoskeleton, but we've also learned in the past that actin filaments make up the cytoskeleton. These are thinner filaments, okay? Microtubules are bigger, whereas actin filaments are much more thinner, and they basically, right under the cell membrane here, they form almost like a drawstring, and they get tighter and tighter and tighter between the membranes that hold the two cells to get the one cell together, but then eventually pinches off and you get two new cells, and it splits the cell into two. That's a cleavage furrow in animal cells. Plant cells do not have a cleavage furrow. Instead, what happens with them is in telophase, at the end of telophase, you start to see these little vesicles that contain cell wall material like cellulose and pectin, and they make a wall, okay, a cell plate. A cell plate is going to become the cell wall that forms between, that separates the two daughter cells from each other, okay? So cytokinesis in plants is different. It's cytokinesis in animal cells, you get a cleavage furrow, but in plants, it's called a cell plate. So first question here, cytokinesis usually, but not always, follows mitosis. And actually that's true for some cells, like for example, the cells that make up your heart, those muscle cells, some of them have not gone through cytokinesis. They've gone through mitosis, but not cytokinesis. So continuing on here, if a cell completed mitosis but not cytokinesis, what would be the result? A cell with a single large nucleus, a cell with high concentration of actin and myosin, a cell with two abnormally small nuclei, a cell with two nuclei, or a cell with two nuclei but with half the amount of DNA. The answer is going to be D, okay? It's going to be a single cell with two nuclei. And in fact, your heart cells are multi-nucleated, meaning like when you look at one heart cell, it actually has multi-nuclei. There's more than one nucleus. So this is question number 3. I did, just for the sake of time, I did remove question number 2. Measurements of the amount of DNA per nucleus were taken on a large number of cells from a growing fungus. The measured DNA levels range from 3 to 6 picograms per nucleus. In which stage of the cell cycle was a nucleus with 6 picograms of DNA? Okay, so we have the cell cycle, right? We have mitosis, we have cytokinesis, we have G1 of interphase, the S phase of interphase, and then we have G2 of interphase, right? So when I see here, fungus, okay, it has anywhere between 3 to 6 picograms of DNA. This tells me it's 3, and then after the S phase, DNA is duplicated, so it would be 6. So then you would have 3 picograms of DNA in G1. By the end of the S phase, you would have 6. You would still have 6, and then you would still have 6 until cytokinesis is when the two new cells would have 3 each, right? So then 6 picograms, not G0, not G1. S, you know, any point during the S phase, it's going to be between 3 to 6. So the answer is going to be G2. Okay, for sure, it's going to have 6. In mitosis, that's where it's going through cell division. So they are going to be separated. And so the answer is going to be G2. A group of cells is assayed for DNA content immediately following mitosis and is found to have an average of 8 picograms of DNA per nucleus. Those cells would have blank picograms at the end of the S phase and blank picograms at the end of G2, okay? So again, this is very similar to the other question where you have mitosis, cytokinesis. This is going to be G1, this is going to be S phase, this is going to be G2, and it looks like it's 8 and 16, right? So it's going to have 8 at the start. Okay, it's going to have 8 here, and then by the end of the S phase, it's going to have 16 all the way through the G2, right? And then it's going to split up. So I'm just going to draw it like this, 8, and then it's going to split in cytokinesis, 8, and then 8. So those cells would have blank picograms at the end of the S, right? So it's going to be 16, and blank picograms at the end of G2, 16. So the answer is going to be D. Okay, next we're going to talk about the cell cycle control because, you know, when you are developing as an individual, as a living organism, you really need to control like which cells are dividing, when they're dividing, how they're dividing, okay? So it's very important for us to, you know, regulate when and where cells are dividing. As little embryos, you know, the cell cycle is very short. It's less than 20 minutes because it's dividing and growing into a little, you know, organism and little fetus, which is going to become a baby, okay? Whereas your skin cells are constantly dividing at a regular pace in a 12 to 24 hour cycle. I mean, they divide frequently throughout your life. Again, mentioned liver cells. Liver cells, even though they're in G0, they do retain the ability to go back into the G1 and then the S and G2. If you, remove part of your liver, then it would grow back. Whereas your muscle cells and brain cells, they're always in G0. Once they reach maturity, they go into the G0 phase where they are permanently in G0 and you do not make any more brain, nerve, or muscle cells. So the cell cycle is controlled by several checkpoints, and we're going to be learning about 3 of them. There's more than 3 checkpoints, but the cell cycle is controlled by several checkpoints. So in this diagram, this is our cell cycle, and you can see that there are 3 checkpoints. There's the G1 checkpoint. It kind of got cut off here, but this is the G2 checkpoint. And then right over here, this is the M checkpoint, M for mitosis. These are the 3 that you need to know. There's more than 3, but there's only 3 that we're going to actually learn in this class. These checkpoints are like the stop and go signals. The cell is growing in G1, and in order for it to go through the S phase, it checks to see that the cell will check to see that it has, enough nutrients to get it to go to the next stage. Is it ready to go into the next stage? Is it big enough? Is there enough food available, nutrients available to move to the next phase? G2, at this checkpoint, is the cell ready for mitosis? Okay, and in the M checkpoint, it's actually called the anaphase checkpoint. Is it ready to complete anaphase? Is the sister chromatids ready to separate in anaphase and move forward with mitosis and complete mitosis? Okay, so these are checkpoints that are really key for the cell to divide and grow in the cell cycle. So what are these checkpoints? Okay, as mentioned before, they tell the cell whether or not it's ready for the next stage. Okay, so in the G1 checkpoint, right, can DNA synthesis begin? Is the cell big enough? Does it have everything it needs to move on to the S phase? Okay. And in the M checkpoint, the G2M checkpoint, has DNA completely synthesized? At that point, if there is a go signal, then it's going to be committed to mitosis. And in the mitotic checkpoint, are all the sister chromatids attached to the spindle? Can the sister chromatids separate correctly? So let's talk a little bit about the first checkpoint. This is the very first checkpoint. If the cell receives a go signal, usually the cell signal is going to divide, okay? And we're going to be talking about some of the internal signals that cause it to actually move on to the next phase of the cell cycle. And then there's external signals like growth factors, which we'll be talking about later, but there are growth factors that cause a cell to go through into the S phase. And if the cell does not receive the go-ahead signal, then it will go into the G0 phase, the non-dividing phase, such as nerve and muscle cells are going to be in the G0 phase. If there's no go-ahead signal in the G1, then it's going to go into the G0 phase. So what are these go-ahead signals? These go-ahead signals are usually proteins. Inside the cell, these proteins are called promoting factors. Outside the cell, they could be called growth factors. And inside the cell, usually these kinases are the go-ahead signals, okay? And these kinases, which we talked about before, kinases are enzymes that phosphorylate. And usually when they phosphorylate an enzyme, or usually when they phosphorylate a protein or a molecule, it activates the protein, it turns it on so that it can stimulate or trigger the next part of this, what we call a signal transduction pathway, okay? So the cell's going to get a go-ahead signal, and these signals could be internal or external, and usually the internal signals are going to be kinases that phosphorylate, that activate the cell to go into the next phase of the cell cycle. And we're going to talk about these go-ahead signals in more detail. So what are these kinases? They're called cyclin-dependent kinases. These are kinases are enzymes that phosphorylate, but they depend on cyclins, meaning CDKs, which are called cyclin-dependent kinases. CDKs they are activated only when cyclin is around. So when cyclin, which is a protein, is around, it's going to activate the CDKs, and together they're going to work together and make a CDK cyclin complex. And those are the go-ahead signals for the cell to move on to the next part of the cell cycle. So the CDK cyclin complex, it triggers the cell to go into the next stage. So here's an example of a CDK cyclin complex. In this example, the complex is what we call the MPF complex or go-ahead signal. MPF stands for mitosis promoting factor, okay? So it is what it sounds like. The go-ahead signal at the G2 to M checkpoint, so it promotes mitosis. So when the MPF is formed within the cell, when this go-ahead signal is made in the cell, it tells the cell to go into mitosis. It promotes mitosis. So what makes this go-ahead signal? You need to have two pieces, okay? You need the CDK, which is this guy, and then you also need the cyclin. You know, the amount of cyclin changes throughout the cell cycle. It starts to accumulate in the S phase. So your cell will start to make more and more, okay, more and more of those cyclins until there's a high concentration of cyclin. If there's a high concentration of cyclin, it's gonna collide with the CDK, and they're gonna make a complex. They're gonna make the MPF complex, and that's gonna be the go-ahead signal for the cell to go into the M phase. And then what happens is that the cyclin is going to degrade. You know, the cell is going to get rid of the cyclin, and all you are left is with the CDK. And that CDK by itself is not a go-ahead signal. You need the cyclin and the CDK to come together to form this the complex, the CDK cyclin complex, which is the go-ahead signal. Okay, so this is one way that your cells can regulate the cell cycle. There's another one. Okay, so we just talked about this one. There's another go-ahead signal called APC, and it is what it sounds like. APC stands for anaphase promoting complex. So you also have a cyclin, you also have a CDK, and when they come together, then they tell the cell to go ahead and finish anaphase and complete telophase, complete mitosis, and you're ready to go into cytokinesis. Okay. The APC is only formed when all of the sister chromatids are attached to the microtubules, the spindle fibers in metaphase. Okay. So those were some examples of external signals. The CDK cyclin complexes are the CDK cyclin complexes are internal signals, whereas the external signals would be like growth factors. Okay. Growth factors are proteins that are released by the body cells that stimulates the cell to divide, okay? And usually they are stimulated to divide when there's a lot of space, okay? When there's not a lot of crowding, then the cells will divide to fill in the space. So it's called density dependent inhibition. Like cells will continue to divide if there's space, but then once it's crowded, then they'll stop dividing. Okay, so like in this example, in this experiment, they like removed a couple of cells, and then the cells divided by mitosis to kind of fill in the space. And also, most cells need to be attached to a substrate. They need to be attached to other cells or other types of tissues in order to divide. That's called anchorage dependence. So growth factors, again, they are external signals, and you can see in this picture, when they bind to the receptor protein that's embedded in the cell membrane, it causes a chain reaction events called the protein kinase cascade, where phosphorylation activates molecules within the cytoplasm. And in this case, it's going to send a message into the cell to start making these CDKs and cyclins for cell division. Okay. Example of a growth factor would be PDGF, which is platelet-derived growth factor. These are proteins that are made by platelets in your blood, and it causes cells to divide and make more. And that kind of makes sense, right? Because if you have an injury, then PDGF will be released so that the cells will, regenerate and heal the tissue. Okay. Another example of a growth factor would be EPO. Okay, EPO is short for erythropoietin. I believe that's how you say it. It's also a growth factor that causes you to produce more hemoglobin protein, which is necessary for red blood cells so that you can transport oxygen to all of your cells. Okay, and there's a picture of Lance Armstrong, who was one of the most decorated cyclists from the United States. He won Tour de France like 7 times, but it turned out that he was taking EPO, which is a banned substance, because EPO causes cells to produce more hemoglobin and more red blood cells. And if somebody has a lot of hemoglobin and red blood cells, they have an advantage because red blood cells deliver oxygen. So you would, somebody with more, an athlete with more red blood cells would have an advantage over just your average athlete. So the last topic today is how cell cycle control, when it's out of control, it can cause cancers. Okay, so growth factors can create cancers. These genes called proto-oncogenes. Proto-oncogenes are genes that are responsible for making growth factors, but if these genes are mutated, then they will continually make growth factors. And you saw on a previous slide how growth factors stimulate cell division, right? It stimulates cell division. So if these genes are not, controlled, if these genes are out of control, then they will cause the cell to continually divide and cause cancer. An example of a proto-oncogene would be RAS. When it's being expressed, it activates cyclins, and cyclins and CDKs, when they come together, they are the go-ahead signals for the cell cycle. And so when it's out of control, you can get tumors growing, which is a type of cancer, right? You also have tumor suppressor genes. Naturally you have these genes that, regulate cell division. And so if you have a mutation in a tumor suppressor gene, then those genes are no longer working, and then cells will divide uncontrollably and cause cancer. An example of a tumor suppressor gene would be P53. Okay, so let's talk about these genes. Okay, so cancer is essentially, the failure of cell division control. All cancers that we know of, it's usually due to damaged mutated P53 gene that's not working, okay? Because P53 is so important for the cell cycle to be controlled. So yeah, if you do not have a working P53, then your cell cycle enforcer or the cell cycle control is no longer functioning. So let's see how P53, this regulator gene, actually works, okay? So when DNA is damaged by maybe radiation or some other carcinogen or mutagen, okay, when it changes or damages our DNA, cell division, it stops, okay? And P53, it triggers enzymes to actually repair that damaged DNA. Also, P53 can cause the damaged cell to actually commit apoptosis. Remember, apoptosis is cell suicide. So it can trigger the destruction of the cell, so that cell will no longer be around to reproduce and pass on that damaged DNA. So you guys, P53 is so important because it allows for cells to repair their DNA, but also it stops that cell that is damaged from reproducing. However, if somebody has an abnormal P53 gene, like the P53 gene is, damaged itself, then it will fail to stop DNA being repaired. It will fail to stop that damaged cell from reproducing, and so that damaged cell will continue to reproduce by mitosis and could possibly become a cancer cell. Okay, so P53 is very, very important as it helps regulate the cell cycle and stop damaged cells from reproducing. So how does cancer actually develop? Well, there's six key mutations that have to happen in order for someone to develop cancer. And as you get older, these key mutations accumulate, and that's why you hear of older people getting cancers more frequently than younger people is because, you know, these 6 things eventually accumulates over time. So for example, unlimited growth by turning on growth promoter genes. And these genes, they produce these proteins that promote, you know, the cell cycle. Ignore checkpoints. Like for example, maybe there's a mutation in P53. And so when P53 is mutated, then the cell is going to continue to divide even though it has DNA damage. And so therefore it's going to escape apoptosis. Instead of the cell dying, the damaged cell dying, it's going to reproduce by mitosis and produce more abnormal cells that have these mutated genes. And also unlimited divisions. You know, most of our cells are limited to about 50 cell divisions, but some cancer cells are unlimited and they can divide forever, okay? And also promotes blood vessel growth. So cancer cells can actually promote blood vessel growth so that blood flow goes to the tumor and supplies the tumor with oxygen and nutrients so that it could grow even bigger. And then these cancer cells, they overcome anchor and density dependence, meaning they don't, you know, most cells will stop dividing when they get to a state of density when it gets too crowded, but cancer cells, they don't take that into consideration. Cancer cells will divide and produce tumors no matter how dense it is, packed it is with cells. And they do not have to be growing on other cells. They don't have to be growing on tissue. They could be, they don't have to be anchored to develop, to reproduce. So what causes these, 6 hits or these 6, mutations that causes cancer? UV radiation, chemical exposure, radiation, unhealthy lifestyle like cigarette smoking, pollution, exposure to pollution, age. As I said, as you get older, you accumulate all of these mutations. And then also genetics. There are some genes that make you prone to developing, let's say, breast cancer. Like there's 2 genes, BRCA1, BRCA2, that causes breast cancer in families. There are 2 types of abnormal masses of cells. Like there's a benign tumor. These cells, they usually just stay within that original site of development. Like they're not going to move anywhere, but this tumor is just going to get bigger and bigger, and you can remove it easily by surgery. But there are some malignant tumors that will actually leave the original site and can spread. And when it does spread and form tumors in other parts of the body, we call that metastasis. And metastasis can cause a lot of bodily harm. It could do a lot of damage as they impair the functions of organs throughout the body. All right, well, that's the end of the video. I hope that this video was helpful. We learned about mitosis, the purpose of mitosis, the steps of mitosis, and the cell cycle, and what could happen if the cell cycle is not checked, if it's out of control. And anyways, I will talk to you later. Bye-bye.